Hello students, welcome to 60 out of 60 in KCET Chemistry 2025 series in just 200 days. So today is the most important day and a happy day I can say, because today we will be completing the whole syllabus of PU2. So biomolecules last part we'll be doing today, where we are going to discuss vitamins, nucleic acid and hormones. Okay, so quickly we'll complete, and with this we'll be done with the PU2 syllabus for our KCET 2025 examination. Okay, so we'll be practicing a lot of questions also today. And one more thing, one request, please join our WhatsApp channel. The link of that will be given in the description section. So if you join our WhatsApp channel, it will be helpful for you, because you can get access to all these notes PDF in the PDF form. You can also get access to our mock test, question paper, solutions. Everything is available in our WhatsApp channel. So please do not forget, join the WhatsApp channel. Okay, now let's get started without any further delay towards the last video or the last chapter of PU2. Okay, and after this, do not think that the series is done, we are completed. After this we will start PU1 chapters as well. Okay, so now this is biomolecules. One second. Yeah, this is your biomolecules chapter. Okay, so now if you see part 1, part 2 and part 3, in part 1 we have already covered carbohydrates, part 2 we have done proteins, we have done enzymes, and in part 3 we will be doing today vitamins, nucleic acids and hormones. Okay, so coming to weightage wise, as I told you, every year you can easily expect 2 to 3 questions from this chapter. Okay, and this is very easy to get this 2 to 3 questions, because this is completely theoretical and you need to just remember certain things, make proper short notes, then definitely you will be able to crack this 3 to 4 questions or whatever it is. Okay, fine. Now coming to part 3, vitamins, nucleic acids and hormones. In nucleic acids we are going to discuss chemical composition, structure and functions. Okay, before that we'll also discuss briefly about the types of nucleic acid. Okay, now coming to the first thing, that is vitamins. What are exactly vitamins? How to define vitamins? So definition nowhere it is asked, but just to understand, so vitamins are the compounds. So first thing you need to know that these are organic compounds required in small amount. Okay, so they are required in very small amount to perform specific biological functions for normal maintenance of optimum growth and health of the organism. So to perform certain specific functions for normal growth, for maintenance of the human body, we need these vitamins in small amount. Okay, next coming to what are the classification of vitamins. Okay, classification we'll see. So vitamins are of two types, one is fat soluble and one is water soluble. As the name suggests, for fat soluble means it will be dissolving in fats and oils. Water soluble means it will be soluble in water. Now you can see water soluble, it is soluble in water. Now what exactly you have to remember here is which are the vitamins which are soluble in water. Vitamin B and C are soluble in water. Now it should be taken regularly in the diet. Why? See, because these vitamins are soluble in water, when we when we excrete, okay, so these vitamins can be lost from the body. So that is why it is very important to consume these vitamins regularly. Okay, now coming to fat soluble. As I told you, as the name suggests, it is soluble in fat and oil and insoluble in water. Okay, now what are those vitamins? Examples of fat soluble: A, D, E and K. Okay, please remember, B and C water soluble, A, D, E, K. A, B, C here, then D, E and K are fat soluble. Next, they are stored in the liver and adipose. Okay, next coming to classification. Next, this is your deficiency and diseases. This is very important. Okay, here is the name of the vitamins. Okay, next here from which sources we can find them. Okay, this sources is not that important, but the symptoms or the deficiency disease is very important from the competitive exam, in your PU exam, everywhere, in the match the following or some other type. This question has been asked every year. Okay, so please prepare this table thoroughly. Okay, vitamin A, if there is a deficiency of vitamin A, then we get xerophthalmia. What is the meaning of this? hardening of cornea of the eye, and another symptom is night blindness. Okay, so next coming to vitamin B1, you should know the chemical name here of vitamin B1, thiamine. Okay, sources you can see, if it is deficiency is there, then we get beriberi. Then vitamin B2, riboflavin, and here we get cirrhosis, or what is the meaning of that? Fissuring at the corner of mouth and lips. Okay, then you can also have digestive orders, burning sensation of the skin. Okay, so that is vitamin B2. Next we have vitamin B6. Okay, that is pyridoxine. So here the deficiency disease is convulsions. Then you have vitamin B12. Here that is pernicious anemia, that is RBC deficient in the hemoglobin. So please remember the chemical name of this one. Okay, B1 is thiamine, B2 is riboflavin, and B6 is pyridoxine. Then vitamin B12 is there. Then we'll go to vitamin C. Its chemical name is ascorbic acid, and that is all of you, many of you might be knowing, many of you might have faced this disease also, scurvy. Okay, then vitamin D, your bones is affected, that is your rickets. And next one you have vitamin E, which causes increased fragility of RBCs and muscular weakness. Next vitamin K, that is your blood clotting time is increased and you lose a lot of blood if you are deficient of vitamin K. Okay, so that is all about vitamins. What two things only you need to know: water soluble and fat soluble, which are the vitamins, deficiency diseases and their sources, and the name of the chemical name of certain vitamins. Okay, now we'll go to the next important and I'll say the bigger segment of today's lecture, that is nucleic acids. Okay, so first, as I told you, types we will discuss. So nucleic acids are of two types. Okay, one is DNA and one is RNA. Okay, what is DNA? DNA is deoxyribonucleic acid. Okay, and what is RNA? RNA is ribonucleic acid. Done. So now we will study about them in detail in our upcoming slides. Okay, so first you should know there are two types of that is, one is DNA and one is RNA. Okay, now coming to the chemical composition of nucleic acids. Okay, we'll discuss what is this nucleic acids made up of. So nucleic acids have three important components. Okay, first is a pentose sugar. Nucleic acid is made up of pentose sugar plus, plus it has to be a phosphoric acid, and then it has a nitrogenous containing heterocyclic compound, or we call it bases. So there are three parts of a nucleic acid, that is pentose sugar, phosphoric acid plus bases. Okay, these bases are what? Nitrogen containing heterocyclic compounds. Okay, so clear? Nucleic acid is made up of three things. First is a sugar, pentose sugar, phosphate group, and then a nitrogenous base. Okay, now this sugar varies in case of DNA and RNA. Okay, so in DNA what we find, see, this pentose sugar is different in RNA and DNA. Okay, now how is it different? So if you check, okay, in case of RNA, which one we find? The sugar that is present is called as beta D ribose. Okay, so I'll write the name. In RNA, which sugar is found? Beta D ribose. Okay, this point you should remember. What is the sugar present in RNA? That is beta D ribose. Next, what is the sugar present in DNA? DNA sugar is beta D 2-deoxyribose. Beta D 2-deoxyribose. Now what is this one? See, why is it called deoxy? So here if you see, there is a OH group present at the second carbon, right? Now here you see there is only H. So there is a removal of oxygen, so that is why we call it beta D2-deoxyribose. So this one is present in DNA and this one is present in RNA. This point you should remember, it can be asked as an difference between DNA and RNA. So in RNA the sugar present is beta D ribose, in DNA the sugar present is beta D2-deoxyribose. Okay. Phosphoric acid PO4 3-, that is present in both of them. Now coming to this bases, what is this bases? As I told you, nitrogen containing heterocyclic compounds. Now these bases are also different in case of DNA and RNA. So in DNA, okay, what are the bases we find in DNA? We find 4 types of bases, okay, A, adenine, thymine, guanine and cytosine. In case of RNA, what happens? The rest of the 3 are same, okay, 3 of them are same, but one is different. We get adenine, okay, uracil, then guanine and cytosine. So what is the difference here? In case of RNA, there is no thymine. In place of thymine, we have a new nitrogenous base, that is uracil. Clear? Now is it clear? Let me revise it. Nucleic acids are made up of 3 components. There should be 1 sugar, pentose sugar, 1 phosphate group and 1 nitrogenous base. Now this pentose sugar is different in case of RNA and in case of DNA. In RNA it is beta D ribose sugar, in case of DNA it is beta D2-deoxyribose. Now I explain why it is called 2-deoxy. Next we have phosphate group common to both of them in DNA and RNA. Then comes the nitrogenous bases. So in DNA we have 4 types, that is A, T, G, C, adenine, thymine, guanine and cytosine. In case of RNA also we have 4, but the only thing is the thymine is replaced with uracil. This much clear? Okay. Next we'll go to something about, okay, one second, yeah. Next we'll discuss about the structure, okay, more in detail. So there is something again called as nucleoside, okay. Now first of all, if you think like you have seen carbohydrates, okay, carbohydrates are made up of monosaccharide units, proteins are made up of amino acids, right? Similarly, nucleic acids are made up of nucleotides, okay. So what I am saying is, see here, nucleic acids composition, nucleic acids are made up of something called as nucleotides. Nucleic acids is made up of nucleotide. Now each nucleotide has these 3 components, okay. So here I should write, better it is if I write nucleotide, okay, that will be more relevant, right. So nucleic acids are made up of 1 unit, a monomer, a polymer of another unit called as nucleotide. Each nucleotide has 3 components, that is pentose sugar, phosphate group and your nitrogenous bases. I hope this is clear. Nucleotides, see I can draw one diagram and show you. See, nucleotides are made up, nucleic acids are made up of these small units called as what? Nucleic acid is made up of these small units called as nucleotides. Now each of these nucleotides is made up of 3 things, that is your sugar, pentose sugar, right, then phosphate group and then bases. Then this is the composition of nucleic acids, okay. Now then what is this nucleoside? See, I told you nucleotide has 3 components, pentose sugar, nitrogenous base and phosphate group. So if you exclude this phosphate group, what are 2 things left? The pentose sugar and the nitrogenous base, and that is nothing but your nucleoside, okay. Now again if I draw and show you, see this is a nucleic acid. This nucleic acid 1 unit is called as what? Nucleotide. Nucleotide 3 parts are there, that is your pentose sugar, okay, pentose sugar, then you have base and then you have phosphate ion, right. Now these 2 units are called as nucleosides. Addition of these 2 units alone is called as nucleoside. Nucleoside plus phosphate, you can say nucleotide. Clear? Okay. Now let us see the next part. Okay. So unit formed by the attachment of a base to 1' position of sugar is known as nucleoside. So that means attachment of base, that is nitrogenous base, to the sugar. Okay, at what position? At 1 position. The sugar position, carbons in the sugar as named as 1', 2', 3' and 4'. So the base is attached to the 1' carbon of the sugar, and that is called as nucleoside. Okay, I'll show you the diagram also. Now when nucleoside is linked with phosphoric acid at 5' position of sugar moiety, we get a nucleotide here. Okay, so what happens? Just imagine, sugar group is there, at 1' we have nitrogenous base, and at 5' position we have phosphate group. So when this nucleoside binds to the phosphate group, we get nucleotide. Okay. Next, nucleotides are joined together by phosphodiester linkage. So this is, we have studied in all the 3 biomolecules. Carbohydrates, it was glycosidic linkage which connects the 2 monosaccharides. Similarly in proteins, the amino acids are connected by which bond? Peptide linkage. And the nucleotides are connected with phosphodiester linkage between the 5' and 3' carbon. So this, when I'll show you the diagram, it will be more clear. The phosphodiester linkage is between 5' to 3' carbon of the pentose sugar. Okay, so now we will see, one second. Okay, so now you see, this is a, what is this? You think and tell me. This is a sugar. Okay, we say that sugar are pentose sugars, right? So this part, okay, this is a, yeah, so this is a pentose sugar. Carbon number 1, 2, 3 and 4 and 5. So at 1' carbon, this is a base attached, right? So this is a sugar plus base, nitrogenous base. So this is nothing but your nucleoside. Okay, side, remember, S-I-D-E, nucleoside is this one. Now if you see next one, this is again 1', 2', 3', 4' and 5'. To the 5' carbon, we have attached a phosphate group. So all the 3 components are present here. So this is one base, you have one base, you have the sugar moiety and you have the phosphate group. So this particular composition is called as your nucleotide. Okay, so now many nucleotides form one nucleic acid. Okay, there are many nucleotides combined by which linkage? By phosphodiester linkage to give you nucleic acids. Okay, fine. Now I'll show you how the phosphodiester linkage takes place. Now you see, this is one sugar moiety, this is one nucleotide. Okay, this is another nucleotide. Here 1', 2', 3', then you have 4' and 5'. Similarly here it is 1', one second, yeah, this is 1', this is 2', then you have here 3', here 4' and here 5'. So now what happens, there is a linkage between the phosphate group attached at the 5' group and the OH group present at the 3' group. So what I am saying, the phosphodiester linkage, we have this one, right? Phosphodiester linkage. How is it formed? So phosphodiester linkage is formed by the phosphate group attached at which position? 5' position with of nucleotide 1. Okay, and we have another nucleotide 2 where there is OH group, okay, attached at 3' position. So there is a bond between here, these 2 particles, and this bond is nothing but your phosphodiester linkage. Okay, now you can see here in the diagram, this 5' phosphate group and this 3' OH group has combined to give you this phosphodiester linkage. Okay, so now how common, there's a, what I can say, a very simple structure of nucleic acid. You can see we have first sugar, base and phosphate group. This whole unit is called as what? Nucleotide. Okay, nucleotide. Similarly, sugar, base, phosphate, this group continues, and what is this bond? This bond is phosphodiester linkage. Clear? So now again, if you want to little revise and clarify this part, okay, I'll do it once again with diagram, simply, like with very simple, we'll try to understand. So we have like this 3. The nucleic acids are made up of monomer units called as nucleotides. Okay, many nucleotides form nucleic acids. Now each nucleotide has three parts. One is your sugar, then you have base, okay, and then you have phosphate group. Okay, sugar, base and phosphate group. Now this sugar and phosphate, okay, this sugar and phosphate is called as nucleoside. Okay. Next, these two nucleotides are connected by this bond which is called as phosphodiester linkage. Clear? Now, this this base is attached to 1' carbon of sugar, and this phosphate group is attached to the 5' carbon of sugar. Okay, and this nucleotide bond is formed between 5' of one and 3' of another. Okay, so that is all about the structure of nucleic acids. Next, if you see, like we have primary and secondary tertiary structure in the protein, similarly nucleic acids are also having primary structure and secondary structure. So in primary structure, what happens? It is the sequence of nucleotides in nucleic acid. Like you remember, primary structure of proteins is the sequence of amino acids. Similarly, what are the different nucleotides present in that particular nucleic acid? The sequence of that nucleotides is nothing but your primary structure. Okay. Now coming to secondary structure. Secondary structure is two nucleic acids wound about each other and held together by hydrogen bonds. In secondary structures, we have two nucleic acids coiled around each other. Okay. Now the two strands are complementary to each other. Now I will explain what is this complementary. See, suppose we have different nucleotides and the nitrogenous bases are present. Suppose in the first strand we have A, T, G and C. Okay. Then in the second one we will have T, A, C and G. So always A forms bond with T and C forms bond with G. So if A is present on one strand, the opposite strand of that at the same position of A, it should be T. Okay, that is called complementary to each other. Next, A forms hydrogen bond with thymine, whereas cytosine forms hydrogen bond with guanine, ATGC. Okay, so now you can see the structure of this DNA. This is how these are, you can see these are your nitrogenous bases here, right? They are forming bonds, hydrogen bonds are present, which acts as a backbone for this particular structure. Two helical coils are there, they are wounded around each other, and how are they held? What is the force that is keeping them? The hydrogen bonds between the nitrogenous bases. Now how many how these bonds are formed? A will formed with T, G will formed with C. That is complementary. Okay, so now suppose you have you have a strand like this, you have a DNA strand like this, and here different nucleotides are present, on that different bases are present. Suppose you have this is the strand of one DNA chain. So there will be another DNA chain which is coiled around this one. So what will be its nucleotides? The sequence of these nucleotides in the another DNA chain you can easily guess, because it is a complementary to each other. So here must be C, here must be G, here must be T, here must be A, and here also must be A. Okay. That is about the sequence of that is the sequence of secondary structure. You can easily find out the nucleotide chains in the another DNA molecule. Okay. Now coming to functions of nucleic acid. So if you know, so functions, that stores your genetic information, it is responsible for protein synthesis. Okay. Now there are some types of RNA also that you just need to know the names. We have mRNA here, that is called as messenger RNA. Okay. Then you have tRNA, that is transfer RNA, and then you have rRNA, that is ribosomal RNA. Okay. What is this? M stands for messenger RNA, T stands for transfer RNA, and R stands for ribosomal RNA. Okay, clear? Fine. So next we'll go to hormones. Okay, so hormones, what is there? Some important points about hormones, let us see. They are intracellular messengers, okay. They act as messengers in the human body, and they are secreted by which glands? Endocrine glands, and they are poured directly into the bloodstream. So these are secreted directly into the bloodstreams and they act as messengers, okay. Now coming to how many types of we have this hormones. We have categorized basically on 3 types. First is your polypeptides, then you have amino acids, and then you have steroids, okay. Examples of polypeptides you can remember, insulin, endomorphin, and glucagons, okay. Examples of polypeptides: insulin, endomorphins, and glucagons. Amino acid derivatives example, you have epinephrine, norepinephrine, and then thyroxine, okay. Epinephrine, norepinephrine, then thyroxine. Last one, steroids, you have estrogen, androgen, and testosterone, okay. So these are the 3 types: polypeptides, amino acid derivatives, and your last one is steroids. Under first one, polypeptides, we have 3: insulin, glucagons, and endomorphins. Then under amino acid derivatives, we have again 3: epinephrine, norepinephrine, and thyroxine. Then steroids, estrogens, and androgens. Now you need to remember their functions, okay. So we will discuss these hormones and their functions, okay. So okay, now if you see here, glucagon is which type of this is a polypeptide form, right? It is used to, or its function is to increase the glucose level in the blood, to increase the level of glucose. Now insulin role is to maintain the sugar level in the blood. Epinephrine and norepinephrine, they respond to external stimuli, okay. So their form, so see these two together, this tries to increase, this tries to decrease. So these two hormones together maintain the blood sugar level, okay. Now if you see in epinephrine and norepinephrine, they respond to external stimuli, okay. Next coming to, so this was what? Amino acid derivative, right? This was polypeptides, okay. Next, thyroxine is also amino acid derivative. So here some important points about thyroxine. It is produced by thyroid gland, right? We just studied that hormones are secreted by endocrine glands. So thyroxine is secreted by thyroid gland. Next, iodinated derivative of amino acid tyrosine. What is the structure? So here there is an iodinated derivative of tyrosine, okay. Tyrosine is an amino acid. In there, if the H is replaced with iodine, we get this one. Next, if we have low concentration of thyroxine, okay, if this hormone is less in our body, then we suffer from hypothyroidism. What are the symptoms of that? Lethargy, obesity, okay. Low concentration of iodine will be there in the blood. Now if you have more concentration of this thyroxine, if the thyroxine concentration increases, then it is, our situation is called as hyperthyroidism, and the so consequence is that, or symptoms you can say, enlargement of thyroid gland, okay. So if low concentration is there, it is called hypothyroidism, and you feel obesity and lethargy. And if the thyroxine concentration is more, excess, then we call hyperthyroidism, and the consequence is again enlargement of thyroid gland, okay. Next, if we see the functions of our steroid hormones. So steroid, what do you mean by this steroid hormones? So steroid hormones are produced by adrenal cortex and gonads, okay. So those hormones which are released by adrenal cortex and gonads are called as your steroid hormones. Now in steroid hormones also we have glucocorticoids and mineral corticoids which are synthesized by the adrenal cortex, okay. Adrenal cortex releases which type of hormones? Glucocorticoids and mineral corticoids. Now if you see the function, so glucocorticoids synthesized by adrenal cortex, just as I told you, what is its function? It controls the carbohydrate metabolism, okay, the function, the role of carbohydrate in the body, modulate inflammatory reactions, and are involved in the reactions to stress. How a person reacts in the stress. You might have seen some people are very cool and calm, and some people like they become nervous and all. So that all depends on your hormone, that is glucocorticoids. Okay. Next one is your mineral corticoids. As the name suggests, mineral, it controls the level of excretion of water and salt by the kidney. Okay. Salt and kidney excretion of water and salt by the kidney is regulated by your mineral corticoids. Clear? So we had three levels, three types of hormones. Polypeptides, under that we have seen we have glucagons, insulin, right? And your glucagons increases the blood levels, insulin decreases the blood level. Next we have amino acid derivatives. There we have seen epinephrine and norepinephrine and thyroxine. So epinephrine and norepinephrine responses to external stimuli, and thyroxine is one which you know low concentration, high concentration we discussed. The last one was steroid hormones. Under steroid hormones again we have like these are those hormones which is released by adrenal cortex and gonads. So adrenal cortex releases glucocorticoids and mineral corticoids. Glucocorticoids release maintains your carbohydrates, metabolism, inflammatory reactions and reaction to stress. Mineral corticoids maintains about the excretion of water and salt from the kidney. This is clear. Now here there is something you should know about addition disease. Okay, when is this addition disease caused? It is when you have malfunctioning of adrenal cortex. If your adrenal cortex is not working properly, then we have a symptom or we have a disease called as additions disease. Now what is the symptom of that? So you feel hypoglycemia, you feel weakness, and you have increased susceptibility to stress. Now if you see, if adrenal cortex is not functioning properly, then glucocorticoids will not be released correctly. If glucocorticoids will not be released, then your stress management will be less. So that is why you will be more prone to stress. In small incident stress things, you may get stressed more. Okay. So addition disease is the malfunctioning of your adrenal cortex. Okay. Now we will, I told you steroid hormones are synthesized by adrenal cortex and gonads, right? So we will see these all glucocorticoids and mineral corticoids are synthesized by adrenal cortex. Now what are gonads? Which hormones are synthesized by gonads? So we can see here. So next first hormone is your testosterone. It is synthesized by gonads. Okay, it is the major sex hormone in males. Okay, and what is it is responsible for secondary sexual characters in male, right? Deep voice, facial hair, general physical constitution. So whatever when the male attains puberty, whatever is the secondary sexual characters that is developed in a male, that is due to this hormone, that is testosterone. Okay. Next we have estradiol. This is also a female sex hormone, and what is its used or what is its function? Development of secondary female characteristics and participates in the control of menstrual cycle. Okay, so when a female attains puberty, they also develop some certain secondary sexual characters, and that is what is your that is what is regulated by this estradiol. Okay, next you have progesterone. Progesterone is another female sex hormone, and what is its function? It is responsible for preparing the uterus for implantation of fertilized egg. So you know, when in a woman there is a fertilization of egg and sperm, so a zygote is formed, right? And the zygote is implanted in the on the uterus. So for that some lining, or you can say the uterus is prepared for the implantation of zygote, and that is controlled by progesterone. Okay, so these were the different hormones and their functions. So I have in the NCERT book, it is given in a paragraph form. I have made it in the table form so that you can remember them easily. Okay, so now we will practice some questions. So this was a question that I asked in my previous class, very simple question. What makes up the primary structure of proteins? So primary structure of proteins is made up of amino acids, right? Next, DNA fingerprinting is now used in forensic lab to identify the racial group, to determine the paternity. So all of this. Okay. Next, which of the following is not present in a nucleotide? So if you remember, nucleotide is made up of nucleotide has three parts: sugar, then you have nitrogenous base, and you have one phosphate group. right? Now this sugar is what? Sugar is made up of nitrogenous bases adenine, guanine, cytosine and thymine. Tyrosine is an amino acid, which is not the correct answer. So option number D here. Next, adenosine is an example of. So okay, now adenosine is an example. So if you see, we have only adenine. Okay, adenine is nitrogenous base. Okay, but when we have adenine plus the sugar group, okay, then we call it adenosine. So that is what? A nucleoside. So option number A, correct answer is nucleoside. The vitamin which is water soluble and antioxidant. So if you remember, vitamin C and B are water soluble, but vitamin C is antioxidant. So correct answer is option number C here. The chemical name of vitamin B1. The chemical name of vitamin B1 is thiamine. Okay, option number D is the correct answer here. Okay. So next one homework question is there. So read the following statement carefully. Albumin is a simple protein amino acid. Alanine contains an acidic side chain. Insulin is a hormone. Muscle contains protein keratin. Point out the wrong statements in the above set of statements. So just read these statements. We have completed the whole chapter. So now you can find out which is the wrong statement and you have to correct them in the option. Okay. So do not forget to comment down the answers, and I hope that you have liked this whole series. We have done with the PU2 sections. Now I want you to comment down that which next video on PU1, which chapter you want for PU1, from where you should start. So that, like the first chapter, some basic concepts of chemistry, it is very low weightage, right? But it is a very basic and a most important chapter, most important concepts are there, right? So high weightage topic is chemical bonding. So I want you to comment down that which chapter of PU1 you want to start with. Okay, we'll be doing all the chapters, but definitely I want to take your opinion about which chapter you want to start. Okay, so thank you for watching, and please remember your target. We need to hit that perfect 60 score in KCET 2025. We are working hard, and it's now your turn to work every day for this goal. Okay, so thank you for watching once again, and please subscribe the channel, and see you soon in the next episode of this series.